Ladies and gentlemen, welcome back to another episode of the Untold Hypothesis, brought to you by the Department of Life Sciences, Kristu Jayanti University. Today we have with us a guest from the University of Florida, Dr. Daniel Czyz, Associate Professor from the Department of Microbiology and Cell Sciences. Welcome, sir.
Thank you for
It's a
having me.
pleasure to have you. And so, since you've been here for quite a while right now, so could you tell us how it feels to be here?
Yeah, I've been at Kristu Jayanti University since Monday early morning. I arrived here around 5 a.m. on Monday, and from the early start, we had a lot of meetings. The schedule has been very busy, but I did get a lot of chances to walk around, explore the campus, see all the facilities, meet with students, interact a lot with students, with faculty. It really feels like a very close community, almost like a family, which is very unique for a university of this size with 60,000 students and 700 faculty. It really feels like family. It's been a fantastic experience.
Right, and we did have the opportunity to interact with you and explore our frontiers in sciences, and we actually thank you for that opportunity. So, Professor, we're just going to go through a series of questions, right? So it will range from personal level questions to a little technical questions. So we can begin with it, right? So just to start off with, how did you get into this particular field within microbiology? How did you begin to explore antimicrobial resistance?
Yeah. So I started research early on as an undergraduate student, and I did my undergraduate at the University of Illinois at Chicago in biochemistry. And I did research early on as an undergraduate. Then I did my graduate studies in molecular biosciences and biotechnology with Professor Rick Morimoto at Northwestern University. And then decided to do something completely different, where I joined the Department of Microbiology at the University of Chicago, and that's where I was first really exposed to microbiology. So I did a postdoc with Howard Schuman and Sean Crossan, and I was working on bacterial pathogenesis and developing host-targeted therapeutics for common and not common, but for intracellular bacterial infections: Coxiella, Rickettsia, Legionella, Brucella, and so on. So when I then, and as a graduate student, I was studying the regulation of the heat shock response, and I was working a lot with disease models of neurodegenerative diseases in C. elegans, using C. elegans as a model organism. So then, as a faculty, I combined these two research areas, and I started exploring the effect of bacteria on these neurodegenerative disease models. But in addition, I continued my postdoctoral work, which was developing non-traditional approaches to target antibiotic-resistant bacteria. So as a faculty right now, I do 50% of research and 50% teaching. I teach antimicrobial resistance to both undergraduate, master's, and PhD students. I have about 250 students each semester, and the other 50% of my time I spend with doing research. So we're developing these non-traditional approaches to target antibiotic-resistant bacteria, mainly concentrating on silver nanoparticles, bacteriophages, and I continue work on the host-targeted therapeutics. And the other half of my lab works on the we're studying the role of gut bacteria on neurodegenerative diseases: Alzheimer's, Huntington, Parkinson's, ALS, and so on. So it was with respect to antimicrobial resistance, it was really a continuation of my postdoctoral work that I started at the University of Chicago. And I develop a lot of these projects and ideas also through teaching, both the laboratory section and lecture section.
Well, that's quite the multidisciplinary approach I can see
It,
there,
yeah.
from pathology to neurodegeneration. That's very interesting. So a little bit of technicality here. So when we talk about antimicrobial resistance, so we are aware that there are certain antibiotics that target specific microbes in different ways, right? Some target the cell walls, while other affect the synthesis of genetic material. So how can this serve as a basis for resistance in bacteria? So since there are different targets, how can they adapt to resistance across targeted molecules?
Yeah, yeah. Uh, So antimicrobial resistance, it's out there. It's in the soil. It's in the ocean. It's present within high density polymicrobial populations, right? Because looking at the population of microbes, it's a constant war. One microbe is trying to survive and thrive on limited resources, and another microbe says, "No, I'm gonna get rid of the competition by producing these antimicrobial compounds, antimicrobial peptides. So I'm going to kill the competition." So now the competition says, "Well, we're going to develop these specific resistance mechanism to resist the agent that is targeting us." And given that bacteria really multiply rapidly,
Right.
some multiply every 20 minutes, there is so much room to develop these random mutations that will confer resistance to just about any drug or any antimicrobial compound. Now, if we increase the selection pressure in a clinical setting such as hospitals, that potentiates, so that effect increases more, leading to widespread antimicrobial resistance. In addition to the vertical transmission of antimicrobial resistance, such as point mutations, for
Right.
example, in the ribosome, and so on. We also have horizontal transmission, which is the
Yeah.
horizontal gene transfer, and that's more serious because it allows bacteria to transport these plasmids in between, within species, but also between species, and sometimes between different genera of bacteria, which creates really selective environment for the development of these new resistance across different bacterial niches.
So if we are to target microbes within, let's say, a human system, right? Apart from antibiotics, are there any other way or any emerging sources that could actually target them?
Yeah, the issue with antibiotics is that they have a lifespan, and it's estimated that an antibiotic, single antibiotic, has about 50 years of clinical use. After 50 years, it just becomes expired because microbes become so resistant to it that it becomes useless in the clinical settings. So, given that most antibiotics were discovered during the golden age of antibiotics during the 50s, 60s, and 70s, add 50 years to that, and now we're approaching the expiration of all of these antibiotics, and that's exactly what is happening. So now what do we need to develop new antimicrobials, antibiotics specifically? If we do, microbes will eventually develop resistance. On average,
Right.
it takes about 7 to 8 years for a microbe to develop clinical resistance from the start of using the antibiotic in the clinical setting. So just imagine that if we develop novel antibiotics, the microbes will eventually develop resistance very rapidly. So even the new antibiotics that are being developed right now, they're not put out there as first therapy. They're put off the shelf, and they're sort of a reserve for being used as last reserve antibiotics because we need to conserve these novel antimicrobials. So what do? We have to invest resources and research and money into developing other approaches. And these non-traditional approaches, well, they could be, for example, what I work on are silver nanoparticles and bacteriophages and these host-targeted therapeutics. So for silver nanoparticles, what we're working on are these fine spheres that are between 1 to 3 nanometers in diameter. And unlike silver ions, which is positively charged and can have multiple targets within a cell, and that also increases their cytotoxicity to the host cell, silver nanoparticles are much less cytotoxic. They're more structured and less cytotoxic. But what we're found is that these silver nanoparticles can potentiate the efficacy of antibiotics, a very specific class of antibiotics called aminoglycosides, by over 60-fold. So now imagine that these aminoglycosides are antibiotics that are uh, commonly used in neonatal medicine. Now, they cause ototoxicity, which is irreversible hearing loss, but they also cause nephrotoxicity, which is kidney damage. Now, the physicians have to weigh in the consequences. Do we risk for an infant to lose hearing or have kidney toxicity, or do we lose the life of a person, right? So sometimes the benefits of giving the antibiotic and with these adverse side effects outweigh the risk of losing a patient. Um, but if we could combine silver nanoparticles with aminoglycosides, we could lower the dose. So now antibiotics are non-toxic, but we're still within the therapeutic window, and that would be a deal breaker, right? I mean, that would be amazing in the field of clinical field to treat these resistant infections, and that's exactly what we see in a laboratory settings. We could potentiate the efficacy of these aminoglycosides with silver nanoparticles at lower without causing any toxicity, but retaining the efficacy
Therapeutic
of
efficacy.
these antibiotics. Um, so that's one example how it could be sort of adjuvant therapy to restore sensitivity of microbes to antibiotics that these microbes already became resistant to. On the other hand, we also work on bacteriophages. And even though phage therapy, bacteriophages have been around for much longer than antibiotics, they're still not being used in the clinical settings. There's still need we still need much more work to do to really develop well-controlled clinical trials to understand how bacteriophages function in the context of a polymicrobial community and in the context of an actual infection. Sometimes these phages work excellent in a test tube, but when we translate them into animal model, that adds another level of complexity. But phages have been very successfully used across animal, agriculture, environment, and of course in human medicine as a compassionate treatment, compassionate care in humans. So there are these different approaches, non-traditional approaches that really give us the different alternative strategies how we could effectively target these bacterial infections that normally are just extensive pan-drug resistance, and there is nothing else that antibiotics can do. But with the utility of these non-traditional approaches, we could actually restore susceptibility, and we could specifically target microbes. In the example of bacteriophages, so going back to antibiotics, for example, broad spectrum antibiotics, they could target any bacteria on our body and in our body, not only the ones that are causing the infection. So if we eliminate our protective gut microbiome, now we're more predisposed to infections and colonization by other more pathogenic bacterial infections. We're making this void space is now introducing pathogenic bacteria. And that's, for example, what's happening with Clostridioides difficile, C. diff, which is it's associated with overuse of antibiotics, clearing off the commensal gut microbiota, and then C. diff takes over, and that causes very severe consequences on the health of the individuals who have these increased abundance of C. diff infections, and these are extremely difficult to treat. So really employing bacteriophages, which allow us to specifically eliminate only that pathogen that is causing the infection without disrupting the other protective gut microbiome, it also gives us the advantage, right? And it protects us against the adverse reactions or adverse effects from antibiotics that we don't think about, and these are the long-term consequences. The the immediate action of an antibiotic is to clear off infection, but these antibiotics have a role in targeting our protective gut microbiome. And if that happens, then we see long-term consequences years from now or decades from now that could have a detrimental effect on our health. And we don't think about that long-term consequences. We think about treating infection within 7, 10, 14 days, but not what's going to happen within 5 or 10 years, or 20 or 30 years. And that sort of overlaps with the other half of my lab. What we're working on is the bacterial pathogenesis in neurodegenerative diseases, when we're learning that these gram-negative Enterobacteriaceae, enteropathogenic bacteria that are colonizing individuals asymptomatically, And these bacteria are not supposed to be part of our commensal gut microbiome, yet a lot of individuals asymptomatically carry these bacteria. And over time, these bacteria increase inflammation and contribute to this chronic inflammation. And eventually, the hypothesis is, and all the data points towards their pathogenicity in these neurodegenerative diseases. So using bacteriophages to specifically eliminate bacteria that are only causing the disease without affecting the protective commensal microbiome, that gives us another advantage of using some of these non-traditional approaches. There are also other approaches, for example, host-targeted strategy, which is much less developed. So in cancer, personalized immunotherapy, CAR T, that's emerging as a very effective treatment for certain cancers. Why can't we borrow the same concept in infections with infectious diseases? And the idea would be to extract stem cells from an individual, and we've done these we've done these experiments in animal models where we extract bone marrow stem cells. We differentiate these into phagocytic macrophages in a dish. We activate the ability of these phagocytic macrophages to kill bacteria, and then we inject them back into the infected animal. And with that approach, we could clear off any bacterial infection. In addition to that, once the bacterial infection is cleared, these animals become resistant to any subsequent exposure to lethal exposure to bacteria. So there are several different approaches how we could target these bacterial infections in addition to antibiotics that are expiring.
Based on the trends, right? So, do you think we are approaching a true, post-antibiotic era? Do you think we're heading there, or we're still stuck in something that could potentially be detrimental to mankind in the long run?
Right. I think we're already at the post-antibiotic era. As I talk with physicians, the emergence of extensive drug-resistant microbes and pan-drug resistant, which are bacteria that are resistant to every single antibiotic, and that's becoming very common for Pseudomonas, Acinetobacter, Klebsiella, E. coli, Salmonella, Shigella. We see this extensive and pan-drug resistant across almost every individual bacterial species. And they really have emerged early on, 1991, 1993. This is the first emergence that we have seen with pan-drug resistant Pseudomonas aeruginosa. And over the span of just 30 years, we saw emergence of extensive and pan-drug resistant of almost every clinical known or clinically relevant pathogen. So we're already at the post-antibiotic era right now. And this is something also that government officials are aware of. They're developing these national action plan. They're developing special panels on lowering the use of antibiotics across agriculture. I myself, I am involved in the National Institute on Antimicrobial Resistance Research and Education, NIAMRI, which addresses antimicrobial resistance from a One Health perspective. We're really tackling the problem both from human health, environmental health, animal health, and plant health. And really, given the interconnectedness of bacteria and communication and interconnectedness of all of these environments, we can't think about antimicrobial resistance as a single silo problem, a healthcare problem. This is a massive problem that spans throughout One Health, and then this is how we have to approach it. But whether it's resistant in agricultural setting or in humans, we're already past the
Antibiotic
era.
So the majority of studies that have been done, apart from the antibiotic-based therapy, so do they remain largely preclinical, or are they entering the clinical phase of tests?
Right. So we are developing new antibiotics, but it's not new classes of antibiotics. What we're developing right now are iteration of what is already known. So all the classes have been already discovered during the golden age of antibiotics. Actually, the last class of antibiotics was discovered in the late 1980s. Since then, we have not discovered a new class of antibiotics. And the reason for that is probably that we have exhausted bacterial targets, because there is only a fixed amount of bacterial targets that we could target with small molecules. It's possible that we could develop novel targets and novel mechanisms of actions for these antibiotics, but it haven't happened yet. There were some potentially new candidates, but they fail in later development because of toxicity, not only to bacteria, but also to the host. So one requirement is that there has to be a therapeutic window which allows the drug to kill bacteria without cytotoxicity to the host. And that's very challenging. And a lot of drugs, most drugs actually fail at that stage. So
So you're telling me that antibiotic repurposing still holds good with respect to the target?
yes, so we're unlikely that with the repurposing of antibiotics we're going to find new targets. But what we're doing is we're changing the structure of existing antibiotics to overcome the already resistance that has developed, and that's where that, for example, that's common for beta-lactam antibiotics, where we have meropenem, carbapenems, cephalosporins. All of these beta-lactam based antibiotics have been modified in a way that it increases side chains and it increases the molecular structure just a little bit, that it becomes more potent antibiotic, and it overcomes the resistance mechanisms, which in the case of beta-lactam antibiotics, most of them are beta-lactamases or extended spectrum beta-lactamases, which there's thousands of genes coding for these beta-lactamases. So again, it's an evolutionary war, right? We're developing an iteration of a new beta-lactam antibiotics, and then the nature comes up with another beta-lactamase that will still cleave that antibiotic after years of its use in the clinical settings. Really, the development of novel classes, discovery of novel classes is not there. It's an iteration and manipulation of already existing structures that are available on the market.
So, Professor, something just very general. Okay, so we know, we are aware that certain bacteria have shorter regeneration times and reproducing times. So one thing I want to know is that given that a certain selection pressure is applied, how fast will it take for a particular strain to develop resistance towards it?
Yeah, and this is a really good question, but it's really sometimes it's strain specific, it's bacteria, it's species specific, and it really also depends on the surrounding environment, right? When it comes to horizontal gene transfer, the selection pressure can actually increase the transmission of a plasmid that carries the resistance from one species to another, right? So that could really spread very rapidly. When it comes to vertical gene transfer, where introduction of mutations, I mean, we could see the development of resistance in the lab just within a couple of days of growing resistant of growing bacteria in the presence of a selection antibiotic. I mean, in theory, it could only take one cycle, right? And if that one cycle is the right cycle to develop that mutation, then it only takes one cellular replication to give rise to that specific mutation that will confer resistance to a particular antibiotic. Of course, a single cycle is not going to cause the mutation, but given that bacteria multiply every 20 minutes, let's say, on average, and they could enter exponential growth, and we could have 1 times 10 to the 8 to the 9 of bacteria within a small tube or within the context of a whole body. That gives a lot of statistical power to develop these mutations across any genes that are falling within the biological target for these antibiotics.
So is there any kind of issue that would arise if we were to transition towards for example, the silver nanoparticle based therapy? Are there any possibilities that they could develop certain kind of resistance, maybe if not against it, but at least a defensive kind of state to protect them from it?
Right, great question. And I think No matter what we come up with, bacteria will always develop resistance because it's an evolutionary war. We cannot eradicate bacteria. If they wouldn't develop resistance, we would eradicate completely these bacterial species, and that's not going to happen, right? Bacteria will always develop resistance. The advantage that comes in using these non-traditional approaches or novel approaches is that we could use them in combination with antibiotics. So if we have a resistant strain that has specific mechanisms of resistance, if we use it in combination with another drug or adjuvant or bacteriophage, we could actually synergize together with antibiotics to kill that bacteria despite of them being resistant to antibiotics. And that's a common thing with what is seen, for example, with bacteriophages. So a lot of receptors, or quite often the receptors for bacteriophages at the cell surface of bacteria are efflux pumps. So now, if we treat antibiotics, or if we treat the bacteria with specific antibiotics, they start to overproduce efflux pumps. Now, if these efflux pumps function as receptors for bacteriophages, so now bacteriophages will start to infect these bacteria more readily. So now what do bacteria do? Then the bacteria will start to decrease the expression of these efflux pumps or mutate those efflux pumps. So now the bacteriophage cannot bind to them, but at the same time, it becomes susceptible to antibiotics, right?
Right.
So it's a combination therapy really that gives us the advantage and potentiates the efficacy of antibiotics. I don't think any of these alternative strategies will ever be used as a single monotherapy. Antibiotics are essential molecules, and even if bacteria become completely resistant to an antibiotic, it's not the end of the game. That is because we have put in it on average it takes 10 to 20 years to develop a single drug, and it could cost easily up to over $1 billion to develop a single drug. Now that's a lot of time, that's a lot of resources. Is it better to develop new antibiotics, or is it better to develop approaches that will restore the susceptibility to that antibiotic, right? And given already all of these resources that we have put into development of antibiotics, I think we have to concentrate more on strategies that will target the resistant mechanisms so we resurrect the susceptibility of bacteria to these drugs.
That's an interesting take on it. And seeing these adjuvant therapies, so basically, we are focusing on a synergistic approach, right?
Right.
So, and is it conclusive that there is no other kind of monotherapy that could actually target this antimicrobial resistance?
Well, there are, but it would not function as a standalone monotherapy. So, for example, beta-lactam antibiotics are often administered with clavins, which are a class of antibiotics that look like beta-lactams, but they have very little antimicrobial activity. What they do is they sequester the function of beta-lactamases. So now beta-lactamases bind to these fake beta-lactams, which are clavins, and they titrate them away from the actual beta-lactam that then inhibits the peptidoglycan formation.
So they're like analogs.
Right.
Right.
Yeah.
Structural analogs.
Structural analogs that they do not have functional antimicrobial property themselves, but they're mimicking antibiotics, so they could sequester these resistance mechanisms away from their actual job to destroy the antibiotic that is targeting bacteria. But alone, these adjuvant therapies, they can't be used as antimicrobials. They have to be in combination. And I think when we look across different antimicrobial strategies, the most effective therapies are the ones that come in combination. Because even when we look at metabolic inhibitors, sulfamethoxazole, for example, it always comes in combination where we're targeting several different enzymes, several different steps of the folic acid folate synthesis pathway, and that decreases the ability of bacteria to develop resistance if the pathway is targeted from different sides.
That's interesting, Professor. Actually, there's more to it than meets the eye right now. That after speaking to you and interacting to you, that's what we get to know. So, so now from a general perspective, what would you say? Actually, you gave us some pretty interesting tips during your presentation on how to build a successful research career, and so could you please reiterate that here so that our viewers can see and find out?
Yeah, absolutely. I think step number one is to really get involved in research, right? And if students are very serious about having a research career, they have to start doing research as early as possible. Uh, Ideally, it's on early ages of their undergraduate education. If that's not possible, then definitely masters, because then it's going to be much more difficult to get into a PhD program without any research experience. Um, Getting into a PhD program is already very competitive, right? So, uh, the how would the faculty who are evaluating a student know that a student is a good fit if they've never been exposed to research, right? There is no evaluation of their skills. But if a student, even if it's volunteering, right? So if a student finds a lab and starts to volunteer, and even if it's just general laboratory tasks or assisting another graduate student or a postdoc or a research scientist, or doing something tedious, but you're in the lab environment, that already gives you that exposure. And eventually, with time, you become familiar with the terms, with the terminology, with procedures, different methods, and it opens other opportunities, right? You build up your CV, you build up your resume, which then makes you a better candidate for the next step of your career, for the next step into advancing in your laboratory position into maybe a paid position, right? And I think there are other opportunities. If there are no opportunities or limited opportunities within the university or within one place, uh, seek out any other laboratories around vicinity, around town, which if one seeks, they'll find, right? But definitely get involved in research as early as possible, and sometimes research can be translatable. So even if somebody is really interested, for example, in biochemistry, but they're doing something tedious with in some other field, um, take a step back and look larger at the bigger question of what what the student is doing, and really understand the impact of the research. What I'm finding with students is that a lot of them, laboratory research is not always very exciting, right? You could be doing repetitive things over and over and over again, and then at one point, students might lose interest in it because they're doing just one thing 100 times a day. But then, if you take a step back and look at the general question and the general contribution that the student has to the entire field, it really makes sense and it makes a great impact, right? And then you're learning uh, uh, transferable skills that can be applied to other areas of research.
So, I would like to end this with one last question. So, let's say on a hypothetical note that we, to a great extent and largely, solve this issue of antimicrobial resistance. So, where would the shift and focus turn towards? Which other problem would the field of microbiology largely focus on after antimicrobial resistance?
Great question. And I think the next obvious issue would be viral infections, right? And we put much less emphasis into viral infections just because the spectrum of drugs, even for viral or fungal infections, is not as rich as it is for bacterial infections. So even though it's still related to AMR, viruses have always been much more virulent than any bacteria. And really focusing on developing these communicable diseases when it comes to rapidly progressing viral infections. For example, what we have seen with COVID was a fast moving pandemic. within 1 year, within months, it shut down the entire planet. But within a year, we developed vaccines, effective vaccines, and now we could function, and it's still around, but it's not as big of a risk as it was at the beginning. It moved very fast. AMR is a slow-moving pandemic, which is much larger scale than COVID. So we would still have to concentrate on diseases when it comes to microbiology and infectious diseases. There are, of course, other issues with plastics, right? Pollution, microplastics, which when we're now learning, they could contribute to antimicrobial resistance. They could contribute to neurodegenerative diseases. It's very quite novel emerging areas. So they do also affect microbes and the efficacy of antibiotics.
Well, so thank you, Professor. It was great interacting with you.
Yeah, thank you.
And ladies and gentlemen, with that, we come to the end of this episode. For more such episodes, stay tuned to the Untold Hypothesis.